Welcome to all CBSE students, especially 11th standard students. In 11th standard, Unit 3 deals about cell biology. It deals about cell biology, cell structure, cell divisions, such type of aspects. In that unit, the chapter 11 explains the cell divisions, cell cycle, mitosis, meiosis. In that section 1, 10.1 about cell cycle, 10.2 about mitosis M phase, 3 significance importance of the mitosis, 4 meiosis or reduction division, 5 significance of meiosis, importance of the meiosis. Now let us take up the cell cycle. Cell during the cell divisions, the events follow a cyclic pattern. That's why it is called cell cycle. While working on the details of the cell cycle, 3 biologists have got the Nobel Award. In 2001, L.H. Hartwell, Tim Hunt, and P.M. Nurse. With these 2 scientists, I have personally contacted them. Hartwell, Hunt, and P.M. Nurse for working out the details of the cell cycle. Now, let us try to understand the events in the cell cycle. Cell divides both in unicellular organisms, multicellular organisms, prokaryotes, eukaryotes, lower organisms, higher. In unicellular organisms, invariably it is reproduction, either maybe asexual reproduction or sexual reproduction, like Chlamydomonas, like Euglena. In multicellular organisms, the divisions are 2 types, mitotic or equal divisions. They are always meant for growth. Whereas, second type of divisions or meiotic divisions, production of gametes. Meiosis is a part of sexual reproduction. So, meiosis division mainly results in the formation of male and female gametes. So, it is important in sexual reproduction. Cell division cells duplicate their genetic material before they divide, ensuring that each daughter cell receives an exact copy of the genetic material, that is DNA. Now, let us see this cells duplicate, double their genetic material, that is DNA, before they divide, ensuring that each daughter cell receives an exact copy of the genetic material. It is equally distributed and the number will be same as that of the parent cell. So, look at this. For example, in an organism, 20 chromosomes are there. The 20 chromosomes before division, cell division, it becomes a double 40. These 40 are equally distributed between the 2 daughter cells. So, therefore, the chromosome number in two daughter cells is equal. It is also equal to the parent cell. So there is no change in the number. So what should happen at one other time? The chromosome number should become double, or in other words, the genetic material DNA should become double. In other words, the DNA should replicate. Two processes must alternate during eukaryotic cell division. Genome must be replicated in S phase. Genome must be halved, equally distributed during M phase. So this is M phase. Here this is S phase. Then what is cell cycle? The ordered sequence of events, ordered, one after the other, same sequence. Ordered sequence, one sequence means one after the other. Why order? For example, G1 followed by S, G2, M. This is correct sequence. Not G1 is followed by G2. This is ordered G1, S, G2, M. This was asked in several NEET examinations. Remember, G1, S, G2, M. In the life of an eukaryotic cell, from division of a parent cell into two daughter progeny cells, G1, S, G2, M. Phases of the cell cycle: mitotic phase, karyokinesis, cytokinesis. Suppose if you take M phase, it is the shortest period, only 10% of the cell cycle. For example, if a cell takes 100 minutes for the division, complete division, it takes M phase, mitotic phase, that means anaphase, prophase, metaphase, anaphase, telophase, even the cytokinesis. For all these phases, it takes only just 10 minutes. Whereas interphase, at one time, it was not given importance. People thought that it is a resting stage. There are no important events. The important phase is M phase. But now it has been identified, recognized that interphase is the most important phase of the cell cycle. G1, S, G2. Longest period, 90% of the cell cycle. G0 phase, quiescent phase, or sometimes resting phase, not in the cell cycle. So when there is no need for the cell divisions, the daughter cells come out of the cell cycle or take rest or still remain active. G0 phase. So G1, S, G2, M. Events in G1, S and G2 phases. G1 phase, first phase of the cell cycle. It corresponds to the interval between mitosis, it is between M phase and S phase, DNA replication. So what events do occur? During G1 phase, the cell is metabolically active. In what way? During this phase, the cell synthesizes all the materials that are required for S phase. S phase, DNA replication. For DNA replication, there are so many proteins, so many enzymes, so many the raw materials are required. All those are synthesized, kept ready for S phase. Continuously grows but does not replicate its DNA. It keeps all the materials ready for DNA synthesis, but DNA is not synthesized at this stage. S or synthetic phase, DNA replication, the amount of DNA becomes double. the amount of DNA becomes double. But remember, chromosome number remains same. Only DNA becomes double. But there is no change in the number of chromosomes. Internally, inside the chromosome, the DNA becomes double, replicates, but the chromosome number remains same. G2 phase, proteins are synthesized and preparation for mitosis. Mitosis includes prophase, metaphase, anaphase. So many materials are required, so many enzymes are required. All those materials are kept ready in G2 phase while the cell growth continues, because one cell has to become two cells in mitosis. So that means everything should be kept ready for the chromosome divisions and cell division. Quiescent phase. See the dictionary, quiescent. So non-dividing stage. It is called non-dividing stage. Daughter cells resulting from a may exit cell cycle, may come out of the cell cycle, become inactive. Suppose there is no necessity for the cell divisions. So cells unnecessarily do not divide. Stop. So the resulting daughter cells come out of the cell cycle, but still active. Metabolically active, but do not divide. However, the inactive cells may again become active, enter into the cell cycle. They may again enter into the cycle. Inactive cells, as long as the cell divisions are not necessary, it remains inactive stage, quiescent stage. But suppose there is an urgency, the cell divisions are necessary, the inactive cells again become active, enter into the cell cycle and initiate the cell cycle. Problem, suppose onion root cells contains 16 chromosomes, then how many chromosomes in G1 phase? 16. S phase chromosomes 16. DNA becomes double, but chromosome number remains same. After M phase, again 16. Then DNA content of the cells. So the difference between the two problems, here asking chromosome, here DNA. G1 phase remains same. After S phase, it becomes double. At S2 phase also, it becomes double. DNA content becomes double, but here it remains same. Then this is an important diagram of the cell cycle. G1, S, G2, these three stages together known as interphase. M phase, P for prophase, metaphase, anaphase, telophase. You see, it is a very short stage. All the events take place within the short time. This is the longest period. So what do happen during G1 phase? Proteins and RNA synthesis necessary for DNA replication. The next phase is DNA replication, DNA synthesis. Then S phase, DNA synthesis. G2, protein and RNA synthesis necessary for all these stages. Then after the M phase, the cell may go into the resting stage, quiescent phase. But whenever the necessary arises, again it may enter into the cell cycle. This is a resting stage. Now the cell cycle events are controlled by some regulators, some proteinaceous materials. They are called cyclins and cyclin dependent kinases. Here this phase is controlled by cyclin E, cyclin dependent kinase 2. These are regulated by cyclin A, cyclin 2. These are regulated by cyclin B, CDC2. Now look at here, a replication completed, DNA integrity is tested. Whether DNA is normal or any during the replication, any abnormalities, any mistakes or any damages, here it is tested. Similarly, here it is tested MPF, mitosis promoting factor, Anaphase promoting complex. They are tested whether everything is normal, then only it is allowed for Anaphase. So these are all checkpoints. At every stage, checkpoints will be there to test whether the events are going normal or not. If they are not going normal, that cell cycle will be stopped there itself. Suppose something is mistake here, DNA synthesis is not proper, then cell cycle stops here. It will not proceed here. Or sometimes that damaged DNA will be repaired, then only it will be allowed to go into the G2 phase. So this is M phase, this is interphase, where cell cycle stages and events. Then how different types of cells are there in plants, animals, human beings? Embryo cycle, the cells in the embryo divide, they take less than 20 minutes to divide for completion of cell cycle. Skin cells divide frequently throughout the life. They take 12 to 24 hours for every one cell cycle. Liver cells retain the ability to divide but keep it in reserve and divide once every year or two. Very slow. They retain the ability to divide. But whenever the necessity arises, they replicate but take more time. Then mature nerve cells, muscle set do not divide at all after maturity. They will be permanently in quiescent phase, resting phase. Remember this, mature nerve cells and muscle cells. Then there are G1, S, G2, F phase. Whether one after the other in an ordered sequence, in the given sequence. So to divide or not to divide? Who tells the cell cycle to divide or not to divide? So if you go into the other aspects, there will be certain signals to divide or not to divide. Look at here. G2, M, G1, S, G1, S, G2, M. G0 phase, non-dividing differentiated state. Suppose immature cell matures and differentiates into parenchyma, sclerenchyma, or like that in plants. Most human cells in G0 phase. Liver cells, as I told you, very late. Then nerve cells, muscle cells, highly specialized, arrested in G0 can never divide. So here, once the M phase is completed, the cell may go for G0 quiescent phase, or again it may enter into this. There is an option here, depending upon the necessity of the organism, whether the new cells are required or not. If the new cells are not required, the cell cycle breaks here, all the daughter cells will be sent to quiescent phase. Or if the cells are required, those daughter cells also again participate in the cell cycle. Then cell cycle events must be highly regulated. So this is an area where the intensive research is going on. What decides the cell cycle, whether to divide or not Right? How is it regulated? Because cell cycle events are closely linked to the cancers. In temporal manner, according to time sequence, correct time sequence, not more, not less. Suppose if S phase requires 30 minutes, within 30 minutes it should be completed. Regulation of the cell cycle is critical for the normal development of multicellular organisms. If an organism, plant or animal or a fetus or a baby is growing, a seedling is growing, the cell cycles are normal, the divisions are normal. If there is a problem in the cell cycle, either in M, G1, S, G2, then there will be loss of control either in the G1, S, G2, ultimately leads to death, apostosis, that is called programmed cell death. The cell is commits suicide in one way. Instead of going with mistakes into the cell cycle, it commits suicide. There will be certain signals to kill the cell that make the cell to commit the suicide. Or cancer, the cell divisions go unregulated. Genetic and molecular studies in diverse biological systems, means in plants, animals, yeast, bacteria, like have resulted in identification and characterization of the cell cycle machinery. How these events take place, how is it regulated? So different organisms, just like Sinorhabdis elegans, there the apostasis has been studied well. Similarly, yeast, Saccharomyces pombe, then bacteria, then human cells, all these. Drosophila. Cell cycle character, temporally ordered events, time schedule. Every event has its time schedule. Within that time schedule, it has to be completed. Irreversibility. Suppose the cell cycle enters from G1 to S, from S to G1, it cannot go back. That is called irreversibility. Then checkpoints. Every phase, at the end of the G1, there is a check. At the end of the S, there is a check whether the events are taking place normally or not. Then positive and negative feedback, then there will be signals, there will be some messages so that these events are going normally or abnormally. Then what ensures smooth and ordered events of cell cycle? So in normal individual, the cell cycle will go in ordered sequence, well regulated. But how this is regulated? Cyclins, two types of molecules, cyclins and cyclin dependent kinases, drive to the cell cycle. There are two. Remember, cyclins, cyclin dependent protein kinases drive the cell cycle. So cell cycle is regulated by cyclins, cyclin dependent kinases. Different types of cyclins are identified. G1, the cyclins that participate at G1 stage, cyclin D. The cyclins that participate at S phase, cyclin E and A. The mitotic cyclins, cyclins B and A. Then CDKs are also at every stage. But interestingly, what happens at every stage, one cyclin dependent kinase. One from this. At every stage. One cyclin dependent kinases. Cyclins, no intrinsic enzyme activity. On their own, they have no enzyme activity. What they do, they bind to CDK, synthesized and degraded in each cycle. So one particular cyclin participates at the beginning of the G1 stage, and it comes to the end of the G1 stage. At the end, it is degraded. Again, new cyclin comes at the beginning of the S. At the end of the S phase, it is again degraded. But cyclin dependent kinases are not degraded. Essential component of CDK activity. If the CDK has to become active, it has to bind with cyclin. Then I will show you. Similarly, cyclins, different organisms, different cyclins are identified. There is a confusion in the nomenclature of these cyclins and cyclin dependent kinases because they have given different names in different organisms. Do not be confused. Then cyclin dependent kinase. Again, I repeat, at every stage, one cyclin dependent kinase. One cyclin dependent kinase. So, like this is cyclin dependent kinase. Initially, it is inactive. But when cyclin binds to cyclin dependent kinase, it becomes active. That's why its activity is dependent on cyclin. That's why the name cyclin dependent kinase. Cyclin dependent kinase. It has the kinase property to transfer the phosphates, but its activity is dependent on cyclin binding. Without cyclin binding, it is inactive. Now, once it binds to cyclin, it becomes active, shows some enzymatic property, kinase property, and cell cycle progresses, for example, G1 to S. Similarly, from S to G2, from G2 to M. So every stage, one particular type of cyclin, one particular type of cyclin dependent kinase. Inactive when it binds to, so we can always, I compare like this. This is engine, this is driver. When driver comes and sits on the engine and starts it, then the engine becomes active. So, for example, there is a problem in cell divisions at one or other stage, then there will be certain inhibitors to stop the cell cycle, because cell cycle cannot progress. It is not allowed to progress with mistakes. So that will be a problem. So what are the P21, P27 proteins? These are inhibitors, cell cycle inhibitors. So this is CDK, another representation, cyclin, a substrate molecule, product is phosphorylated. Then ATP is converted to ADP, one phosphate is transferred to this becomes active. So this will usually function as a transcriptional factor to activate particular gene necessary for that particular event. This is usually a transcriptional factor. These transcriptional factors activate the genes that are necessary for that particular event, maybe G1, S, G2. So here, then once suppose the cell has G1 completed, what happens? These two dissociate. cyclins and cyclin dependent kinases dissociate. So let us go back. So here they are bound. At the end of a particular event, these two dissociate. Cyclin, see, then it is degraded. Cyclins are degraded. Because they are degraded every time they are freshly produced. That's why the name cyclins. Cyclin dependent kinases are not degraded. Phosphorylation initiates cycle events that results in the activation of certain transcriptional factors. These transcriptional factors are necessary for the activation of genes, expression of genes. These transcriptional factors promote the transcription of certain genes, expression of certain genes whose products are required for the next stage of cell cycle. So this is a molecular events. So look at here at G1 cyclin D CDK4, cyclin D cyclin dependent kinase 4, here cyclin E CDK2, cyclin A CDK2, here like this. At every stage these two cyclin dependent kinases. Then checkpoints. At every stage there will be checkpoints where the events are going as expected. Whether the events are going normally, then only the cell cycle. But if there is some problem, some mistake, some damage of DNA or some other problem, then that will be checked by checkpoints. Checkpoints, one checkpoint between G1S, it will check for can DNA synthesis begin. Another G2M, has DNA synthesis been completed correctly? Commitment to mitosis. Then spindle checkpoint at mitosis. Every stage it will be checked. So these checkpoints are necessary when there is a problem, when there is a DNA damage or the things are not going as expected, the cell cycle will be stopped. As I told you, some inhibitors will be there, P21, P27. Cells do not receive signal to exist, cell cycle switches to G4, not dividing working state. Then checkpoints. Suppose if DNA is damaged by X-rays, CDK cell cycles stop phosphorylating their target proteins. That means cyclins do not bind to cyclin dependent kinases, and that complex doesn't the phosphorylate the substrate, and cross-functional factors are not synthesized, and a cascade of events take place. Inhibitors of cell cycle. If the cell cycle is not going normally, there is a problem, big problem, then inhibitors, certain inhibitors will be there. What are those inhibitors? P21, protein 21. Then P53, this is called guardian of the genome, DNA, policeman of the DNA. It inhibits. If there is a problem, then this. Checkpoint control systems. Checkpoints, this I told you, stop or signals will be there at every stage. Then if the cell cycle goes wrong, two events take place. The cell is allowed to commit suicide, that is called apoptosis or programmed cell death. Or if the cell escapes the apoptosis, it goes on dividing without any control. That will lead to cancer. Cancer essentially failure of cell division control. That's why the cell divisions are given that much importance, because a lot of work research is going nowadays on cancers, how to prevent. What control is lost? Lost checkpoint stops. Gene 53 plays a key role in restriction point. If this fails, also the control is lost. Halts P53, policeman, guardian of the genome, protein halts cell divisions. It detects the damaged DNA. P for protein 53, the molecular weight. This detects whether there is any damage in the DNA. That is most important thing. For everything, the DNA should be normal. So if this detects, and if it detects, what it does, it prevents the cell cycle. Maybe allowed to go for apoptosis or unrestricted. Then all cancers have shut down P53 activity. It is policeman, guardian. If that fails, then cancers result. Then let us see a normal cell division. DNA is replicated properly. Mutations occur when DNA replicates. Mistakes in DNA replication. DNA replicates normal. Chemical signals start and stop the cell cycle. At every stage, there will be signals to go further or to stop. Here, in a cancerous cell, these signals are ignored, just like a vagabond boy, a bad boy, who doesn't heed the suggestions, advices of the parents or the friends or the teachers. It ignores the signals, good signals, whether to go or whether to stop. But this heeds the signals. Cells communicate with each other so they don't become overcrowded. So this is another aspect of the cell. With its neighbors, it will be evenly spaced. But here cells do not communicate with each other and becomes the crowded. That results in the tumor. So this is the result of normal cell cycle. This is the result of unregulated cell cycle. Thank you. This is very interesting topic. In NEET, you will definitely get at least one question. Sometimes they ask 2-3 questions from this